डियर स्टूडेंट्स, पिछले वीडियो में हमने बात की थी स्ट्रक्चर ऑफ डीएनए के बारे में, जो कि एक न्यूक्लिक एसिड है। इस वीडियो में हम बात करने वाले हैं एक दूसरे टाइप के न्यूक्लिक एसिड के बारे में, जिसका नाम है आरएनए या राइबो न्यूक्लिक एसिड। यदि हम आरएनए की लोकेशन की बात करें तो सेल के अंदर द मोस्ट ऑफ द आरएनए इज प्रेजेंट inside the cytoplasm, and small amount of RNA is also present inside the nucleus in nucleolar region. And as far as the composition of this RNA is concerned, it is the polynucleotide chain. And when we compare it with the structure of DNA, as we all are familiar that DNA is a double-stranded or double helix chain, while there is only a single chain of polynucleotide present inside the RNA. And obviously the structure of this polynucleotide we already have discussed in the previous video. In this video we'll focus on the structure of three basic type of RNAs which is present in almost every type of the cell. And these three types of RNAs are messenger RNA, also known as mRNA. Second one is transfer RNA, also known as tRNA or soluble RNA. And third one is rRNA or ribosomal RNA. And we will be discussing in detail the structure of messenger RNA and transfer RNA, and as far as the structure of ribosomal RNA is concerned, it is beyond the scope of this class, so we won't be discussing its structure, rather we'll be discussing few of its characteristics, properties. So first of all we come to the mRNA, that is structure of mRNA. mRNA is a longest ribonucleic acid or RNA present inside the cell. It is present in the linear form, and the different portions of this mRNA are as follows. First of all, there is methylated cap region. This is methylated cap region, followed by a smaller non-coding region. It has the start signal, or which is also called initiation codon, and initiation codon is AUG or GUG. Further, it has a long coding region, which is obviously a sequence of nitrogen bases, which is complementary to that of the DNA present inside the nucleus. Here it is followed by a small stop signal, which is known as termination codon, and as far as the termination codons are concerned, this is UAA, UAG or UGA. Further, it has smaller non-coding region, and at last it has poly-A tail region. So this is the simplest structure of messenger RNA as is given in the syllabus or prescribed in the syllabus of NCERT. As far as the quantity of or amount of this mRNA present inside the cell as compared to the total amount of RNA present inside the cell, it forms only the 5% of total amount of RNA present inside the cell. Obviously, it is a linear and the longest RNA present inside any cell. And its main function, as the name indicates, is the transcription, or carrying the information from the DNA present inside the nucleus to the cytoplasm for the synthesis of polypeptide chain and ultimately to the synthesis of proteins. That is why it is called as mRNA or messenger RNA. The second type of RNA about which we'll be discussing over here is pRNA or transfer RNA. Transfer RNA is, as far as its size is concerned, it is the smallest RNA amongst all the three types of RNA, and as far as its amount is concerned, it constitutes about 15% of total amount of the RNA present inside the cell. And as far as its structure is concerned, it is explained by clover leaf structure of RNA, जो आप मेरे साथ यहाँ पर ड्रॉ कर सकते हैं. It has, this is first loop, this is smaller loop, this is the second loop, which is obviously the anti-coding region. Third loop, and this is the region where the amino acids will attach. This is C, C, A and OH region. So this is the region for attachment of amino acid. This is the loop which is responsible for attachment with the ribosome where the synthesis of polypeptide occurs. So we call it as the ribosomal site. Ribosomal site. Here we have anticodon. To this region we call anticodon region. And this is the site for the attachment of enzyme. So we can call it as enzyme attachment site or enzyme site. And obviously this is the site for the attachment of amino acid. And these anticodon are obviously complementary to the codons present on mRNA. And basic function of this transfer RNA is to bring the specific amino acid according to the sequence of nitrogen bases present on the coding region of mRNA to the site of polypeptide synthesis, to which we call as a translation, about which we will be studying in detail in the next class. So these two types of RNA, the structure of which we have discussed over here, are messenger RNA and transfer RNA. As far as the ribosomal RNA is concerned, the structure of ribosomal RNA is beyond the scope of this class, as we have discussed earlier. So here we will be discussing few of its characteristics. Like, ribosomal RNA is the most abundant RNA present inside the cytoplasm of the cell, and ribosomal RNA, rRNA, constitutes about 80% of the total amount of the RNA present inside the cytoplasm of the cell. and its main function is to help the two smaller subunit of the ribosome to attach with each other and to prepare them for the synthesis of polypeptide chains. So in a nutshell, we can say that these three types of RNA are responsible for the process of transcription, that is bringing the information from DNA to the site of polypeptide synthesis, and translation, that is synthesis of polypeptide chains. In next video, we'll be discussing about the comparison or differences between DNA and RNA. Till then, take care.